Dr. Ed Zimmerman, Aesthetic Revolution Las Vegas, and I'm back with Dr. Chuck Meeker, who is a double doc. He's both a PhD and a JD, and we're talking about umbilical derived stem cells and exosomes. We've talked a lot about the regulatory pathways, what they are. Now we're going to talk about how we use them clinically. And so, Chuck, welcome back.
Thank
you so much for being here and making my life a lot easier.
It's an absolute pleasure.
He's kind of like the wizard behind the curtain.
I feel a little bit intimidated with this next segment because I'm not clinical. I know a lot about the application of these products in clinical settings, but you are the clinical expert. So this is my time
we'll work
to
together.
learn from you. Okay, we'll tag team. This will be great.
Okay, so from your point of view, where are we at with this technology, this aspect of regenerative medicine? Let's talk a little bit about where we're at realistically.
Sure.
And we talked a little bit about the federal guidelines and things like that. So where are we at? Where do you expect it to gradually go? This will be our little crystal ball, and then we'll look back in 5 years and go, did we get there?
Right. That's good.
How good were we?
That's good.
And what's the messaging that we should be sending to patients and providers about regenerative therapies like this?
So I would say where we're at right now is an intense level of excitement, and the modalities and the procedural protocols are trying to catch up.
Yep.
Everyone is insanely excited about the preliminary results that they're finding, about the availability of stem cells. So they're trying to develop protocols that they can use to implement stem cell therapy and help their patients get the results that they want. We like excitement. We just like to pull back on the reins a little bit to make sure that we're not getting over our skis and getting ahead of ourselves. We want to make sure that things are being done with a proper cadence, So maybe where you start is, what are we seeing these products used for?
Sure.
What kind of procedures they're being used in? You've already mentioned a couple of them, right?
Yep.
You've got surgical wounds that heal naturally on their own without any attention, but they heal slowly and incompletely. And we want to try and address those. We want to activate the body's ability to heal and regenerate itself. So we use a lot of different modalities, and these regenerative products can help tap into those. So wound healing endeavors.
Yeah, wound healing, everything from hair to skin after resurfacing or deeper chemical peels, things like that, after face and neck lifts, anything that disturbs the milieu where the skin and subcutaneous tissue is tacked down to the underlying tissue, especially if we've rearranged some fat or put in, we've grafted some fat from somewhere else, or maybe a allogenic fat product.
Sure.
These are all things that are becoming part of our armamentarium now.
Yeah.
And we're just having fun with this stuff. So, one of the things that you can comment on, I think from your point of view, is what products are legitimately available.
Yes.
Because your company supplies some of those to us, and we appreciate you.
Great. So one of the most popular products that you'll find on the market is an umbilical cord stem cell product. But there's at least two different types of umbilical cord stem cell products. The first one is an umbilical cord blood-derived stem cell isolate, a stem cell product. And the second is an umbilical cord tissue-derived product that contains stem cells. So we didn't talk about this a lot in the last segment, but the umbilical cord is kind of a tubular conduit.
Yeah.
And it's got a sheath on the outside, and then inside that tube is this squishy, gelatinous cushioning material, affectionately referred to as Wharton's jelly. If you've ever heard of Wharton's jelly, that's what it is. It's the gelatinous material that supports and cushions the umbilical cord. Its major function is to protect the umbilical cord vein that runs through the umbilical cord to make sure it doesn't get crushed or kinked or pinched or damaged in any way, because That is where the literal lifeblood is coming from, is through the umbilical cord vein. Turns out the umbilical cord Wharton's jelly is full of bioactive molecules, growth factors and cytokines, stem cells and exosomes, in addition to natural hyaluronic acid, collagen, elastin, and other extracellular proteins that are outside of the cell. Cushions, yeah. So it serves that kind of function biologically as this lubricating, cushioning, hydrating, and supporting material. And when you purify out of the Wharton's jelly those components, they can serve the same function therapeutically that they were performing biologically. And you remember what it's called when products perform the same function therapeutically that they do biologically. We say homologous use. That's right. This is the homologous use of the Wharton's jelly derived components.
Yeah.
Now, we're not truly using Wharton's jelly. It's not a goopy, gelatinous material that we're injecting into people. Wharton's jelly is the tissue source, and we're isolating or purifying out of the Wharton's jelly the bioactive components that we're interested in.
Okay.
But shorthand in the industry is to say this is a Wharton's jelly product. You have to ask the question, though, Wharton's jelly product, which components of the Wharton's jelly are remaining in this product? For example, you could keep most everything that's in there in a complete Wharton's jelly derived product with all of the stem cells and exosomes and growth factors and cytokines and hyaluronic acid and collagen and elastin and other things, and kind of have a kitchen sink approach, a very robust, bioactive, valuable product. But you could also isolate just the stem cells from the Wharton's jelly and call it a Wharton's jelly product. Might be a little bit misleading or disingenuous. It's a stem cell product. It's a Wharton's jelly derived product. Which one is it? So you have to be astute enough to ask the question. It could have the stem cells and the exosomes. It could have the stem cells removed and just keep the exosomes. It could have the stem cells and exosomes removed and just keep the other bioactives, the proteins and hyaluronic acid and everything. In this case, it's still a Wharton's jelly derived product, but it doesn't have any of the stem cell or exosome components. So make sure you're asking the question, what is left in your Wharton's jelly product that you're selling to me?
Because we have different uses for each of these products.
That's correct. If what you want is the delivery vehicle for natural growth factors and cytokines, you better make sure that the stem cells and or exosomes are in there. If what you want is a natural source of hyaluronic acid and collagen and elastin, you may not need those things. You just want the hydrating, lubricating, supporting function of those extracellular proteins, in which case they can keep all those stem cells and exosomes and you can have the remaining fluid.
So in your products, because you list exactly what's in each product.
That's right.
Which makes it a little bit easier for me to fumble my way through and go, well, if I want to do this, I want to choose some of this product and some of that product.
That's right. So at our company, at Hyogen Medical, we chose not to create a catalog of products with different dilutions and different fractionations.
Thank you.
If we're going to do a Wharton's jelly product, we're going to do a complete Wharton's jelly product. We're going to pull out of the Wharton's jelly all of the valuable bioactives that we can keep in a product, and we're going to deliver to you a high efficacy product. So we didn't see a need to produce a Wharton's jelly stem cell product or a Wharton's jelly exosome product, a Wharton's jelly acellular product with just the proteins. If you want a Wharton's jelly derived product, take everything that's valuable about that Wharton's jelly and use it. So we do have one of those. Our flagship products are derived from the umbilical cord blood. You'll know the reason why is because we see the direct correlation between the fetal growth and development, the regenerative function of these umbilical cord blood stem cells in the womb versus in a therapeutic setting. And we want that. If we can see our patients' bodies responding to umbilical cord blood stem cells the same way that the growing baby responded to umbilical cord stem cells, we'll throw a party every single day. And so our flagship products are a Hyogen CV, a cellular vehicle product that contains the umbilical cord blood stem cells with its exosomes and its growth factors and cytokines. And then for specific uses, we have a product that removes the stem cells and only keeps the exosomes from the umbilical cord blood. So you tell me, why might you want an exosome product instead of a stem cell product?
Well,
Yes.
if I want to get through the blood-brain barrier,
Okay.
then I need something that'll cross it. And stem cells are too big
There
in
you go.
general to cross the blood-brain barrier.
Brilliant. So generally speaking, stem cells are too large, considered to be too large to cross an intact blood-brain barrier.
Yeah.
For that reason, if your target is to supplement the blood with growth factors and cytokines across the blood-brain barrier, you would need a different delivery vehicle. You would need an extracellular vehicle, or EV. So we have a Hyogen EV product that has just the exosomes that are produced by, secreted by those umbilical cord blood stem cells. Are there any other barriers in the human body that you might need to cross that have a size restrictive function?
Well, certainly we see as we get into smaller and smaller vessels that are supplying the healing, the baseline
Yes.
healing into areas where we've done surgery, it may get awfully small for a full-size cell to get there. Even a white blood cell has a problem getting through some of those tributaries.
That's right. So even in injectable applications, some exosomes could really be beneficial in delivering into tiny little places. Fortunately, our Hyogen CV stem cell product contains the stem cells and its exosomes.
Yep.
We do find that purified exosome products are used in topical aesthetic applications as well.
Yeah.
The skin is a fantastic barrier for keeping big things out,
Mhm.
and you can either create or induce microchannels in the dermal layer that are large enough for exosomes to pass through. So if you ever go through an aesthetics clinic that does micro needling applications,
Yep.
if you ever do other topical modalities, you'll find that exosome products are used. Now, let me just
But
stop
not
you
all
for a minute.
exosome products are created equal.
You beat me to the punch. I was literally going to say the words, not all exosome products are created equal. So what did you mean when you said that?
Well, we are deluged with advertisements for every exosome product under the sun. And some of them may be animal, some of them may be mineral, some of them may be vegetable, some of them may be human. And so there's a difference in the products, there's a difference in the pedigree of knowing where they came from.
Yes.
There's a difference in how they're isolated, there's a difference in how they're shipped, there's a difference in how they're stored, and there's certainly a difference in the results we've seen from A to B to C.
Yeah.
Okay, now it's your turn.
Okay, so I love that you brought up plant exosomes.
Yeah.
There was this whole shift recently where a group of companies said these are plant-derived exosomes. What on earth possessed them to do that? Well, I think a couple of different things.
Chlorophyll?
Maybe. I think that they're, you know that the human body has an endocannabinoid system, an endocannabinoid signaling system. And our body produces human cannabinoid molecules that bind to endocannabinoid receptors and activate pathways in the body. But if you want to try and supplement the body with cannabinoids, we don't have a renewable source for getting human cannabinoids. So they turn to plant cannabinoids, phytocannabinoids, and they harvest hemp and other cannabis plants.
Yep.
And these phytocannabinoids, they turn out to be structurally similar enough to bind human cannabinoid receptors and weakly but robustly activate these cannabinoid pathways, and you see people getting results. Okay, so in the stem cell industry, people said, I'll bet you plant stem cells work the same way, and if we can use them to activate the body system. The problem is there just isn't enough research to know if the plant-based factors, the growth factors and things, interact with the human-based system enough. And so my suspicion is that most of the plant-based companies said, well, I can't use human stuff, that's illegal.
Yep.
turns out it's not illegal, but they didn't know that at the
They didn't know how
time.
to do
Everyone
it.
was so scared of their own shadow because
Yeah.
the FDA was shutting down stem cell companies that they said, well, I'll just turn to plant-based. Then I know the FDA will never bother me because it's just a plant-based product. The effectiveness of plant-based exosomes, I'm not even going to comment on. I just don't think there's enough research to demonstrate those.
Maybe we can go across the parking lot to the dispensary. Maybe they know more
But
about
maybe
it.
they know more about
Say,
plant-based
one of those
stuff.
yummy gummies.
Yeah,
Something
very
like
good.
that. So
Say, hey, do you have any of those behind-the-counter exosome products?
yeah.
Probably look at you like you're crazy, but
Well,
fun.
but you know, everything's legal in Vegas, baby.
that's right.
Well, almost.
Well, you
everything.
know what we also saw was we saw this really interesting industry pop up where people were using salmon sperm. Did you
Oh
see
yeah,
this?
yeah.
Right. So this was
That's
another
big.
way.
It's big in Korea.
Yes, it was using a different species, but what they wanted was they wanted that gamete source of all of those bioactive proteins, and they're like, this is a great renewable source. We have salmon farms. It's animal, so it's not going to be regulated the same way. We find huge, bioresponses in cell culture, and so they isolated salmon sperm and they started selling it for all kinds of topical applications. People get creative. They get innovative.
Yeah, but there was something fishy about it. Sorry.
Wait, no, but
Took
I
that
mean,
hook, line, and sinker, but
Yeah.
no. We should go off camera when we do that.
that was
Anyway.
great. No,
Oh.
keep those coming.
Okay.
Um,
All right. Well,
I-
the net result is
Wow, this guy. I got to take
Yeah.
some notes.
anyway. Back to cells and exosomes. So we're using them in a variety of things now, and what might tempt patients to come in and ask about whether we can use cells or exosomes to address a number of issues that we're doing in the clinic now.
Yeah.
But you see, you know, we do what we do, but you see
Sure.
the variety of things that lots of clinicians
Yeah.
are doing. So teach me what else the competition's up to.
Right. So generally speaking, before the population is educated enough to come in and ask for something, clinicians, physicians, practitioners need to be educated enough to know what they're offering. So part of our goal is to make sure that when a client comes in and asks a question, that their healthcare provider has the answer. We're doing this educationally as much as anything.
Yeah.
So for you healthcare providers that are out there, you need to know what it is that is offered in the industry, and you need to know what questions to ask when someone walks in the door so that you know what product you're getting. Not all exosome products are created equally. So when someone says, I've got a great new exosome product, it's found in this vial, and all you have to do is take this lyophilized powder and add hyaluronic acid to it to reconstitute it, and then you've got a shelf-stable or a refrigeratable product that you can use for days or weeks or anything. You need to know the product that you're getting, right? Lyophilization is a vacuum and temperature mediated kind of dehydration process.
Yep.
It's more elegant, but the ultimate result is you get this residue that they can then be in a preserved state that doesn't have the fluids and everything in it.
But what is it when you manufacture that? What happens to the cell walls and the vesicle walls, and the activity of that product when it goes through lyophilization and then is rehydrated in a hyaluronic gel? Well, hyaluronic gel is basically the same stuff that's between the cells of all living things, but in our body it is sterile. When you put it into a bottle and now you put the bottle in the fridge or whatever and go in and out and use it again and again, gotta be careful about that sterility thing. There's some
Sure.
red flags going off here.
Well, let's talk a little bit more about it. So I have not seen any lyophilized stem cell products.
Okay.
We know that the cells in our body have a cell membrane around them, and they are filled with water, and water and isotonic pressure is kind of something that is necessary in order to keep these cells viable, live, and intact.
Yeah.
So I think that everyone can agree if you were to lyophilize a stem cell, you would just flatten it out. And it would
Liquid bilayer would go.
Right, that cell membrane. Exosomes, for those that
Yeah.
don't know, are tiny little lipid bilayer bound vesicles that are formed within the cell and then fuse with and are secreted from the stem cell. Well,
Messengers.
yes, in fact, lots of different cell types in the body produce exosomes. So you need to ask the question, where are the exosomes coming from? You know, animal, mineral, vegetable. Are they human exosomes? And then ask, what is the cell type that they're coming from? They might be harvesting exosomes from a different tissue or cell type.
So
Okay.
you're saying the exosomes from my 9mm clock may not fit in my 308 rifle.
Oh,
You know,
so interesting analogy. No, I would say that the more profound question about the exosome is, what's inside?
what's it doing?
The power is on what's inside of these exosomes. They were formed in the cell in order to contain the growth factors and cytokines from the host cell and then to secrete those out. So if you're getting growth factors and cytokines packaged into exosomes in a cell type that you're not interested in its growth factor and cytokine profile,
Exactly.
then you would not also be interested in its exosome profile. So it doesn't matter what form you're getting it from. I have no interest in even cryopreserved plant stem cells or exosomes because I'm not interested in the growth factors and cytokines that are found in them. I want the growth factors and cytokines from the original source of regenerative human repair. I want those umbilical cord blood stem cells that were circulating through the growing baby's system and delivering those bioactive molecules because I want that mechanism of action happening in my body.
Nothing fishy about that.
Nothing fishy about that. So let's talk a little bit about cryopreservation, right?
Okay. Freeze me, baby.
That's right. Stop. Freeze. I'll continue. The in vitro fertilization industry, probably the place where you see cryopreservation happen the most, right? They'll in vitro fertilize an egg, they'll cryopreserve these, keep them on liquid nitrogen at minus 80 to 180 degrees, and they'll lock in life. They'll literally lock in the capacity of these fertilized eggs to be thawed, transplanted into the womb, and grow into human life. So we know cryopreservation locks in bioactivity in life. So when you talk about a cryopreserved stem cell product, you know that analogously, in analogous matter, these stem cells have a preservation that's unique, that locks in bioactivity. Don't just take my word for it. We sent our cryopreserved product off for independent third-party testing.
Right.
They thawed the product and then they stained it differentially with trypan blue and acronin orange, which are two cellular stains.
Yep.
But when you contrast those, you can really see the difference between dead cells and live cells.
Exactly.
Right? And so when they do the staining and they look at the difference, they see an ultra high concentration of live cells left in the thawed product. That gives us the confidence to know that our cryopreservation doesn't damage cells. We've taken all of this great care to make sure that we're not damaging the birth tissue, that we're not damaging the stem cells, that we're using high quality processing reagents and cryopreservative. If at the end of it we thaw it and we've crushed all the cells with ice crystals and freeze thaw cycles and things like that, it would all be for naught. So we love that cryopreserved biologic products give us the comfort of knowing we've safely protected the valuable growth factors and cytokines inside intact stem cells and intact exosomes. When you lyophilize a product like an exosome, that lipid bilayer can rupture. As soon as it ruptures, those growth factors and cytokines are no longer safely protected inside an enclosed vesicle.
Yep.
Now, does it crush 100%, burst 100% of those exosomes? I don't think so,
No.
right? I think that there is a good number of them that are still intact, but I'm shooting for the best that you can possibly do. And I know that cryopreservation locks in bioactivity and growth factor cytokine function. So that's what we've chosen to do, is cryopreservation.
Well, and you check too, so that
Indeed.
when you do your thaw and you go back and count each of the individual exosomes or each of the individual cells. No, not. I'm sure you use a scanning electron microscope or
we use a
hemocetometer.
and yeah, that's right.
Yeah.
That's right. We use cell sorting equipment that allows it to go through and count dyed cells. Right.
Yeah.
We use trypan blue and acronym orange cellular dyes, and then there are cell counting equipment that goes through and does that in an automated fashion. But we do it manually as well. We take that cell sample and we put it on a slide and we put it under a micrograph, and we have the machine count within a field, within 100 different fields, how many of the cells are live and how many of them are dead, so that we can get accurate counts out.
Sounds like what I used to do in cell biology as an undergrad.
Yeah.
In a 4-degree cold room. Yeah, that wasn't fun. But the neat thing about your product is I know I can reliably tell a patient I'm going to use 10 million or 20 million or 30 million or however many million cells,
Right.
surrounded by all the growth factors, the kinases, in ultra high amounts because you've measured the amount of protein available in that sample. Or I'm going to use 100 or 200 billion exosomes.
That's right.
And, as with many things that we do in our practice, size matters.
Right.
And, this is another area where the number of viable vesicles or cells and all the surrounding milieu that you throw in makes a difference in how the product works.
Yeah, let's talk a little bit more about that. So we've gone to great lengths to make it clear that the activity of the stem cell is not what's important therapeutically. The stem cell doesn't do anything in therapy. It's a delivery vehicle.
Yep.
So when we count the number of stem cells and say there are 10 million stem cells in here, it's not because you're getting 10 million stem cells worth of activity. It's because you're getting 10 million stem cells worth of growth factors and cytokines.
Yeah.
That's the therapeutically relevant portion of the product. So cell count is a surrogate number for how many growth factors and cytokines you're getting in there. So at the very least, you should be asking your stem cell source, your provider, how many stem cells are in your product. In a perfect world, you would ask them the more relevant question, which is, what is the concentration of growth factors and cytokines, your whole protein concentration? The fact of the matter is that test will run you $10,000. And so many companies just won't incur the cost to do the test. They use the cell count as close enough. We just know that it's
proportional
10 billion
to.
That's right.
Yeah, and we do the same thing with IGF-1s versus human growth factors and things like that. We measure proportions.
Yeah, but it turns out not all stem cells are created equal either.
Yeah.
So at Hyogen Medical, with our laboratory that processes the stem cells, we've sent not only our product, but leading stem cell companies' products that we've ordered from them into a lab to do a head-to-head test.
How'd that turn out?
What we found is that there's a 10-fold difference in the concentration of growth factors and cytokines between some products. So we looked at these products and found that ours had 5 to 10-fold higher protein concentration. We asked ourselves why. They're all supposed to be coming from human umbilical cords. Why are our products so high in growth factor and cytokines? In some ways, it's because we're using the umbilical cord blood-derived stem cells, which are delivery vehicles for growth factors and cytokines. That's their biological function. But in another very real way, we've taken great care of the cells. So they haven't died, they haven't burst, they haven't been diluted. We're keeping in that product what we say we're keeping. We've learned from sad experience that sometimes a product that's advertised as having 5 or 10 million stem cells really only has a fraction of that. But who's going to check their work? Who's going to verify that? They just end up believing you. And as long as it gives some benefit, people seem to be happy enough with it. We just don't live that way. Numbers matter, words matter. So we make sure we say the right things and we have the right things in our product.
And it matters from lot to lot, because otherwise when we get it clinically, some of our patients get some benefit, other patients don't, and we scratch our head and go, it's the same stuff we got from the other manufacturer. Why didn't it work?
Right. So now it's consistency that matters also.
Yep.
Yep. It's another reason why in our lab there's an FDA certified piece of hematological laboratory equipment. So we don't have to rely on human hands doing the work, because human hands are never consistent.
They're
from
not
one
reproducible
batch to the next, right? Yep. If you've ever done PRP, if you've ever done PRP and you've centrifuged it and you've spun it, and then you've taken the syringe and you've drawn off the platelet-rich plasma portion and you've breached that barrier and watched your syringe turn pink to turn red, you know it's really easy to get red blood cells into the valuable plasma portion. By the same kind of process, if you were trying to spin and isolate just the stem cells from the umbilical cord blood, you would almost certainly make a mistake and pull some of the red blood cells into the product.
Got it.
Right? So now you've introduced something that's antigenic into the product. It's not going to be consistent from one round to the next. So patients may have reactions. Patients may have a different outcome from one batch to another. With our equipment, we know it's the same thing every time. It's gentle on the cells, it's consistent, the results are the same, and so that gives us a lot of confidence to come to you and say, you know what you're getting when you go with us.
Yeah, and you're very upfront about the use of a cryopreservative, the majority of which gets washed out. Mm-hmm. So we don't have to worry if our patients have a sulfa allergy per se.
Yeah, let's get into that just a little bit too. So cryopreservation, which we love, requires a cryopreservative.
Yeah.
These are molecules that help coat and surround the vesicles to make sure that when the temperature drops down, they don't burst. Okay. There's two major cryopreservatives in this industry. The first one is dimethyl sulfoxide, DMSO. And not all DMSO is created equally. You can get it in a very impure form, or you can get it in a highly purified pharmaceutical grade form, which is what we choose to do.
Yeah.
Dimethyl sulfoxide is incredible as a cryopreservative, as a pharmaceutical grade carrier, all of these things, but there is a small percentage of the population that is sensitive or allergic to the sulfoxide moiety. This is very similar to a sulfa allergy, except that a sulfa allergy is tied to a sulfonamide moiety, and this is tied to a sulfoxide moiety, but the way that it presents is very similar. You might get some irritation, some scratchiness in the throat, some flushing, things like that. Nothing that a little Benadryl can't handle, and the Benadryl is not contraindicated with the stem cells, so it's perfectly fine to say, it looks like you're having a little bit of a reaction to the cryopreservative. Let's give you some antihistamine and help take care of that reaction.
We give it to them ahead of time. And it's interesting because DMSO is used by our compounding pharmacies to help the topical anesthetics that we use in aesthetics have some penetration. The DMSO is a penetrant, and occasionally we'll get someone who gets a little red from it, maybe a little itchy from it. We just take it off. It's not a big deal.
That's right.
So that's kind of cool. But
I will say, for those patients who have a known sulfoxide sensitivity, maybe they're coming back in for their second therapy and they say, remember I had a reaction to the last one. We do have a different cryopreservative that we use specifically in some lots, and that's glycerol. So glycerol is also a great cryopreservative. I don't think that it functions quite as well as DMSO, but it is perfectly suitable, and I've never heard of anyone having a reaction to glycerol. No. So for patients that need it, we have those products as well.
What are some of the other risks, and then we'll get into benefits, that you think of in terms of cell therapy?
Well, if you're not processing the product properly, there's tons of risks,
Yeah.
right? You could introduce contaminants, you could leave communicable diseases, you could, all types of stuff. If you haven't properly screened your donor mother, if you haven't tested the tissue sample, if you haven't done all these things, then you really can't say what the risks are. There is a plethora of risks. But if you've done all of these things and you've checked all the boxes and you know that it's a pure sample and you've sent it off every batch, lot by lot, for independent third-party testing to look for contaminants, infectious materials, communicable diseases and things, and you know it comes back clean and clear, and you've got a certificate of analysis from the
We get a
testing
full
lab to show
pedigree
that.
every time we get a batch of
That's,
cells.
that's
from you.
right. When you have that,
Yeah.
now what are the risks in the product? Okay, so number one, while I would never accuse you of having bad technique, there are some providers that don't take great care in their procedural
Yeah,
implementation.
sure.
Jabbing needles into skin and things like that, if you haven't properly cleaned the area, you can introduce bacteria into and underneath the skin. We've even heard of people becoming septic because some skin bacteria was pushed into the bloodstream and then caused an infection. So that can easily be obviated by using the best practices possible in very clean and sterile environments. The risk can't be taken down to zero, but it can be minimized.
Yeah.
Plus, those accidents can be easily treated for the most part. Most people can be easily treated. You find some redness in the area, you know it's getting sore, you know you've got a little infection that happens. It doesn't mean that the infection came from the product. We've
True.
got a certificate of analysis for the product saying it's sterile, and we've archived samples in each lot to make sure that if 10 years later someone says, I think that product gave me a reaction 10 years ago, we can always go and pull that sample and have it tested again.
Well, that's cool.
Yeah.
And you can't control what the patient does outside of your office either.
Yeah, this is another thing. So adverse reactions, we lump this into a category called adverse product reactions.
Yeah.
But they're not all tied to the product. Sometimes it's tied to patient non-compliance. So if you tell your patient, I just did a joint injection on you, I delivered a portfolio of growth factors and cytokines, those growth factors and cytokines are the same ones involved in your body's response to injury, including controlling inflammation, the right amount of pro-inflammation, the right amount of reducing inflammation, and then they go out and do a major workout and re-injure their shoulder, what's going to happen? Those cytokines are going to do exactly what they're known to do biologically. They'll bind receptors and the body will respond by flooding in a lot of inflammation to help lock down the damaged area. Now the patient might come back to you and go, I have no idea what you did to my shoulder, doc. It's so sore I can hardly sleep. But it wasn't your technique and it wasn't the product that caused the problem. It was patient non-compliance following their discharge from therapy that caused this issue. Even if a patient doesn't do anything non-compliant, there is still a risk that a body responds in a unique way. Perhaps there was an injury that was already there and the body was still actively addressing it, and we flooded it with growth factors and cytokines, and there is an acute inflammatory response. This is not life-threatening. It's easily treatable, right?
Yep.
Sometimes it goes away on its own. If it doesn't go away on its own, we can use, an anti-inflammatory, even a corticosteroid in extreme cases to get that inflammation to go away. Typically, we say corticosteroids and NSAIDs and things are not compatible with stem cells because they mute the inflammatory response system while we're trying to tightly control it with growth factors and cytokines.
Yeah. So we combine low-level laser wavelengths and things like that with it at times.
Sure.
So, but we should talk a little bit about the things that a patient would consider in terms of preparation for or care after their stem cell or exosome deployment or injection.
You know, one of the first things I ever heard of stem cells being used for is in the hungover population. Have you heard about this?
I have.
So they do
The drunk bus.
stem cell IVs for hangovers. And I thought this was the oddest thing I'd ever heard of, but essentially they wanted to do an IV therapy to see if they could get rehydrated and reduce some of the inflammation. They thought stem cells would be good for this because it adds additional factors to the body that the body knows how to use to heal and recover. So they began to do things. And I looked at that and I said, what a waste.
It's an awfully expensive way to go.
That's an awfully expensive way to try and counteract a binge drinking night, right? We have, most of our practitioners counsel their patients to avoid alcohol for the days and weeks leading up to therapy.
Yep.
Alcohol, we know, even in small amounts, is pro-inflammatory in the body.
Yep.
We know that smoking is the same way. We know that seed oils and highly processed foods and refined sugars, they all induce inflammation to a certain degree or another in the body. So instituting hydration in advance, clean eating, avoiding substances that cause inflammation. If you really want to get the best bang for the buck, if you're going to spend this much money on a human cellular based therapy, you should really take a little bit of time to optimize your outcome by doing other things as well.
And maybe not starting your new exercise program right after you got it.
Man, I felt so bad. I did know. I got a call from a doctor, and we had to tease it out a little bit because I was confused. What happened? So he went back to this lady and asked, "Tell me what happened in the first 48 hours." And she literally said, "I felt so good the next morning that I went to the gym feeling like $1,000,000, and now my shoulder hurts so bad. What did that product do to me?" And we're like,
it wasn't the product.
It wasn't the product.
The gym.
Yeah. So.
Yeah. So we always give patients a handout when we start to talk about this, and we're going through the education and the consent, so they know what they're getting into to prepare for and to treat after their stem cell injection.
Yeah, excellent.
So, but patients do come in because there is some interest now, and they say, "Well, doc, what can I use this for?" You know, we had a patient that we interviewed and made sense, and she was already going down the stem cell rabbit hole. So she was telling me ahead of time, "Do you do this?" And I'm interested. But sometimes patients just hear something on the street and they come in. What do we tell them about? And we certainly tell them the stem cell doesn't fix something, doesn't turn into something.
Right.
But it certainly does bring the communications to your own body to help heal itself. But what can we address with cells and exosomes? What's reasonable in this day and age?
So many of you viewers may not understand that the question that you're asking is a tightrope walk between regulatorily compliant language and informative information. Nobody wants to be in a position where they have to tell their patient, "I can't talk to you about what this product does, but we're going to use it on your scalp, or we're going to use it in your injection or your IV infusion." That's not satisfying. It doesn't instill confidence. At the same time, you can't legally make claims about what the product will do. And when I say make claims, what I mean is you can't say that these stem cells address conditions in the body. It's not just treating or curing, it's addressing a condition. So at your clinic, you don't use stem cells for inflammation, stem cells for treating inflammation. You don't use stem cells for treating hair loss. You don't use stem cells for treating joint pain. You don't use stem cells for any number of things that they're being advertised for in some circles, because stem cells don't treat those things. A treatment is a term in the industry that means the FDA has cleared it and approved it to address a condition. And these products have not been approved to address or treat or cure a condition. They have been authorized for homologous use. So you've got to make sure when you're talking about this that you provide enough information to satisfy the patient while always going back to the fact that these are used for homologous use only. So how can you do that? Well, you can certainly say how you will administer the stem cells. We do stem cell scalp injections,
Yeah.
right? That's informative. You haven't made any claim about what the scalp injection will accomplish. You can always say, we do stem cell joint injections. We do stem cell IV infusions. And you haven't made any kind of claim about how it will affect the structure or the function of the body, or what diseases or conditions it treats, or what outcomes they will have. When a patient wants to get into outcomes, you have to stop for just a minute and say, tell me what you're interested in addressing. Let's let them talk for a minute. And if they say, I have low back pain and I think that I have inflammation going on, and we go, good, where does inflammation come from? And they say, well, it comes from the way I'm eating, and it comes from environmental factors and things like that. And we go, great, does your body have the ability to address inflammation on its own? And they'll say, well, I don't know, it's not getting rid of it now. And I go, great, I understand that, but your body is programmed, biologically, evolutionarily designed to address rampant inflammation. It's just not doing it right now. Let's talk about what your body needs in order to address inflammation. And then you talk with them a little bit about the factors that are still in their life that are causing inflammation, and those that are preventing their body from addressing inflammation. And then you say, in addition, your body has an incredible mechanism for regenerative repair and getting rid of inflammation. That process involves turning on a pathway that allows your body to reduce inflammation. Those pathways are turned on by tiny little cytokines that bind to cell receptors to activate the pathway. Then you stop and you shift the conversation and you say, stem cells contain many of those same cytokines that our body is using to activate pathways involved in inflammation. So you see what we did there? It's a little bit of a ballet dance, right?
But it's important.
But it's important. If you say, we're going to use stem cells to reduce your inflammation, you just press the FDA buzzer.
Yep.
If you say, we're going to use stem cell growth factors and cytokines to address inflammation, you just press the FDA buzzer. That's not what they do. Addressing inflammation is something that your body does. It's not something that stem cells do. It's not even something that the growth factors and cytokines inside the stem cell do. So you need to help your patients understand. You need to help your patients understand that your body holds the key to addressing many of its own problems. And that what we want to do as healthcare providers is help your body address its own issues. The way that your body addresses many of its issues is by activating innate molecular pathways that are designed to address that issue. And the way those pathways are turned on is by growth factor and cytokine signaling. Full stop, new conversation. Stem cells contain a wide variety of growth factors and cytokines that are identical to the ones that are found in your body that address these pathways, that activate these pathways.
Yep.
And our goal in delivering stem cells to you is to deliver those natural biological human growth factors and cytokines so that there's more of them in your body.
Yep.
And they go, awesome. And it sounds great to them. And you never needed to say, the stem cell growth factors and cytokines are going to activate your body's molecular pathways and it's going to reduce your inflammation. Because that is saying that the product will affect the function of the body.
Yeah.
When we don't want to imply that the product affects the function of the body, we want to imply that the product is being used in homologous fashion as a delivery vehicle for natural biological human growth factors and cytokines.
That you've lost some of the ability of your body to do that over time with aging.
Yeah, very good. So when we were young, we didn't need stem cell therapy. Why? Because our body was full of all of these growth factors and cytokines. Our pathways were turned up to 11. We were going all the time. As we've aged, those pathways have been turned down People say, oh, as you age, there's telomere shortening. And I'm like, yeah, but telomere shortening is a part of the issue. The real issue is that the molecular pathways have been turned down, that the gene expression and molecular mechanisms are no longer functioning at level 10. And if you can turn the pathways back up, your cells behave like young cells. It's almost like reprogramming. It's not reprogramming. The programming is already there. It's reactivating. It's like turning the volume back up on your cells so that you get that amazing music playing again. And it's going boom, 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 boom. But right, and it's this whole big symphony and everyone's excited about it.
The band's all here.
That's right, the band's all here.
Yeah, and that's why, from a clinical point of view too, we're doing both IV deployments and direct injections where we can,
right,
in order to get the word out.
Yeah.
To where we need to get it.
I'll say something else about IV infusion. This is a little known, I don't want to say it's a secret, it's just a little known fact. Part of the federal regulation for compliant 361 HCTPs, which we talked about last time, is that the product is not for systemic use, right? It doesn't serve a systemic function. And so people say, oh, that means you can't infuse the product because blood goes everywhere, and so that means it's a systemic use. And I say, that's not what it means.
I don't think so.
No, what it means is that you're trying to address a particular condition, a unique acute condition, and that condition is the lack of growth factors and cytokines in the blood. When we do IV infusions, we're not administering a systemic therapy, we're administering an acute therapy.
Yeah.
We're supplementing the blood with natural biological human growth factors and cytokines, because when we review the patient's chart, we say, I bet your problem is you don't have enough growth factors and cytokines in your blood, because if you did, I don't think you'd be seeing these problems. I'd like to supplement you with growth factors and cytokines. The way I'm going to do that.
That's a great way to put it.
The way I'm going to do that is by administering an IV infusion of stem cells that I know are full of growth factors and cytokines. So you see what we did there? We just walked it from back to front instead of from front to back. I think your problem is a lack of growth factors and cytokines. Those growth factors and cytokines, I know what they do. The literature is replete with examples of how growth factors and cytokines turn on molecular pathways involved in regenerative healing. If you're not healing, it's most likely because there's too many things pushing the pathways down and you don't have enough growth factors and cytokines turning the pathways up. So let's address these things, and let's address this and supplement your body with growth factors and cytokines.
How cool is that?
It's very cool.
So there are a lot of things that we can address.
Indeed.
That we won't talk about directly.
Yeah.
And
Let's rephrase that. There's a lot of things that the body can address on its own.
Yeah, that's better.
If its pathways are activated. We know that what activates those pathways is growth factors and cytokines, and we know stem cells contain growth factors and cytokines. So why wouldn't we not want to deliver stem cells into a patient's body?
I think I'd like to sign up for a 24-hour drip to
the last time
correct my imbalances.
The last time you had that was in the womb, my friend. I tell you what, one of my favorite things to ask patients is, you should try this in your clinics too. You say, when was the last time you received an IV infusion of stem cells? And they'll say, I've never received an IV infusion of stem cells. And you say, liar! You have received an IV infusion of stem cells in the second and third trimester of gestation, right?
Yep.
We all did. We've all received IV stem cell therapy. That's how they were delivered from the placenta through the umbilical cord blood and into our growing bodies in utero.
Yep. And when they did your C-section,
yeah,
what you really had was a womb with a view.
Yeah.
Sorry.
Keep them coming. I love it.
So we're having fun, but we appreciate you all tuning in. I think anything else we should cover?
Nope. I think we've come. We talked about optimization by removing things that were pushing down pathways and lifting it. I really did want to address that. The reason why I wanted to address that is I don't think stem cells are a silver bullet, a cure-all.
No.
I think this is a multifactorial, holistic approach to health and wellness. There is a place for functional surgery. So all of you orthopedic surgeons out there that think that the stem cell industry is going to take over your practice, stop worrying about that. The stem cells can never do what you do. They're not construction workers. They're not surgeons. They can't fix those things. What stem cells can do is deliver growth factors and cytokines that aren't in high abundance. And every surgeon should want that. You should all be implementing stem cell therapy in connection with your surgeries.
Every surgery.
Because every human body needs a higher level of growth factors and cytokines as they're trying to heal and recover from surgery, accident, injury, or whatever.
Yeah. In concert with the right amount of sleep, the right amount of nutrition, the right amount of supplements, the right amount of gentle physical therapy, and moving into exercises, all those things.
Totally agree.
And maybe next time we'll talk about peptides.
Let's do it. I'd love to talk about peptides.
Cool. All right. We're done for now. Thanks for tuning in.